Hello, everybody. Can you all hear me?
Yes.
Yes, great. Well, thanks everyone for joining today. My name is Deborah Seiler. I'm your webinar host. I'm with the University of Illinois Extension. And we're very excited for today's speakers. This is that I-Pollinate Citizen Science Training webinar. The projects you're going to be learning about today were really inspired by the enthusiasm and dedication of our volunteers within Extension and all the things that they do. And I think this is going to be a great opportunity to connect with some really interesting research happening here at the University of Illinois. So we'll have 4 guest speakers with us here today, and they are the coordinators and the scientists behind the 3 projects that we'll be learning about. We'll have McKelvy, is the coordinator of engagement, technology, and new media for the Office of Mathematics, Science, and Technology Education here on campus. Michael works on a number of public engagement projects for UI, and that includes, he is the webmaster for BeeSpotter and manages the technical side of that project. Michael says his primary background is in IT, so being able to learn so much about bees has been a real perk. Dr. Terry Harrison is an alum and a former researcher in the Department of Entomology here at U of I in Urbana-Champaign. And he has also been the scientific coordinator and bee identifier for BeeSpotter since it was created by Dr. May Berenbaum, who was also in the Department of Entomology in 2007. And a fun fact about Terry is he actually started his insect research when he was just 5 years old, helping out with his big brother's insect collection for a biology class. But as a professional research, Terry's expertise has been in moths. Dr. Alex Harmon-Threatt is a pollination ecologist and an assistant professor of entomology. In her lab, works to understand patterns in natural environments to help conserve and restore pollinator diversity. Specifically, her lab focuses on bees and how different factors in the environment, like plant diversity, fire, grazing, habitat fragmentation, can affect bee diversity in our local communities. And last but not least, our 4th speaker is Dr. David Zaya, a plant ecologist with the Illinois Natural History Survey, who focuses on plant communities across Illinois and how they change over time. And David has used that expertise as a member of the Illinois Monarch project, where he is working to understand the best environmental conditions for planting milkweed to give monarch caterpillars their best chances of survival.
Mute my phone.
Unmuted.
So today we're going to go over the 3 project options that are part of I-Pollinate. First up, Alex and David are going to talk about the 2 projects connected to planting your own garden test pot, just to look for general pollinators and to look for monarch caterpillars. And then Michael and Terry are going to walk us through BeeSpotter. So with that, I'm going to turn it over to Alex and David. And if you have questions, feel free to put them in the chat box, and we'll have time at the end, and we'll try and go down the list and take any other additional questions that you have.
If people are having trouble hearing, can you just test the volume on your speaker just to make sure that they can hear us okay? Because I know some people are having trouble hearing. Yeah, some people can't seem to hear anything. Mm-hmm. Can everyone just try to make sure that people can hear me before I start talking? Okay, so this is it.
If anyone wants to chime in on the chat box with whether you can thumbs up or thumbs down and whether you can hear.
Okay, all right.
Okay,
all right,
great.
Wonderful. So I'm Dr. Alex Harmon-Threatt. I'm going to start by talking a little bit about the garden projects that are available, and then I'll give it over to David to talk a little bit about his aspect of that. So first, I want to thank Extension and Deborah in particular for getting this kicked off. They approached us in the fall, and there's been a lot of work behind the scenes to try and figure out what we could do. Scientists, if we had people who were interested, and so there's been a lot of work behind the scenes by Deborah, especially, and a lot of the other Extension folks to get us to this point. So I want to thank them for their attention and getting us here. And then I want to thank you guys for wanting to participate in science. There are a lot of benefits of having citizen scientists involved in research, and you guys make things possible that would otherwise not be possible for scientists to do. It really helps us increase the distribution of sampling. Illinois is a huge state, and we can't make it to end. David tries every summer to do so in some of his other projects, but it's really challenging. And so you guys allow us to sample a lot more area more quickly and more efficiently. That helps us increase the amount of data, and having more data just produces a lot more robust results. If it was dependent on just me and my small team here at the university to get end to end, we just would not be able to cover what you guys are going to be able to cover for us. And that's going to help, obviously really help improve the results. It increases access to certain habitats. The first project I'm going to talk about is really based in people's yards. And that's an area that we have a lot of trouble getting access to, because we have to ask every single homeowner, like, can we come into your yards? And so by you guys collecting this data, that gives us access to these habitats. And that really actually helps us to better conserve these organisms. I think both David and I have a lot of interest in giving people information on how to improve conservation efforts, and having access to yards and being able to give people targeted results from their homes and what can be done at their homes is really going to help us improve these conservation efforts. So the two projects that are based on these backyard gardens are the Pollinator Visitation Project and the Monarch Project. Both of these are based on a plot that we're going to walk through how to set that up in a minute here. The Pollinator Visitation Project is a project that started in my lab based here in the Champaign-Urbana area. And we were initially just working in parks and local parks. And this year we're stepping up to the statewide project. And we're really excited to have you guys' participation. And the goal of this project is really to better understand how ornamental plants can provide resources and whether or not they're beneficial for pollinator conservation. And the Monarch Project is really an assessment of monarch distribution, where they're laying eggs, and their population growth throughout the state. We know that Illinois has a very little natural habitat left, and so getting some assessments of what's going on over a wide distribution in people's yards will really help us benefit pollinators in general. So I started this, I dreamed up these pollinator gardens in part because I'm a big time gardener. And when I started trying to make my yard pollinator friendly, most of the recommendations are for native plants. And I willingly did this, and then I very much so got a fine from the city because the plants were too tall. And so I started looking around at other plants in my yard that also had a lot of pollinators on them and wondering, could we use some of these plants to make other recommendations? But there ultimately is very little data on ornamental plants. And so then as part of this project with Extension, David and I and my lab tech, Katie Berry, came up with this design. And really we owe a lot of the thanks to Katie because she did a lot of the heavy lifting on this to try and figure out what are some common, commonly available and commonly planted ornamental plants that might be good for pollinators or that we just don't have very much data on. And then within the design, we also included a milkweed species, and that obviously is not an ornamental, but it is a plant that we wanted to have as a focal plant in people's yards. And so the list here, these annual species are all, you don't have to get all of these. We wanted to give people options based on your location and what's available in your local garden centers. And any examples that are given of varietals, that you do not have to get those. We just were trying to give you an example. We just were like, we would like you to have the yellow zinnia and the yellow marigold, and those are examples, but you don't have to try and even look at the varietal. If it looks like a yellow marigold to you, we'll be happy to have you planted in your yard. And so each of these, someone asked recently if it had to be oval. It does not have to be oval. We just wanted to give you a kind of limited amount of space. So if you want to put it in a square orientation, that's fine, or rectangular. The goal is just to get these 20 or 24. What?
24.
Yeah.
Yeah, just the 6 varieties.
Yes. The 6 different, so we wanted to just get these 6 different annual varieties that you can put into this arrangement, or any real arrangement, as long as you're grouping the plants that are of the same type. We just would like them to have some kind of a grouping of those of that type, and that's partly because it'll make it easier to collect data on them in the future. So if you have them grouped together in some way, and you know these different varieties, milkweeds really must be on the edge, because if you're going to be looking at the milkweed, looking for monarch eggs and larvae on them, you want to be able to have good access to them. The others can be a little deeper in, because you should be able to see what you need to see without getting super in close on them. So this is just a design, and we didn't talk about this, but we have Katie designed a really beautiful website. It has all of the information on the garden setups. That's and should be really easily available. And so anything we go over today is also on the web. And so this should be pretty easy. So once you get your, once you've gone to the garden center, figured out what plants are available, and you get those guys in the ground, what we're asking you to do is then to register and tell us what is in your garden. Because we gave you multiple options, we actually need to know what was in your, which ones you were able to acquire, and to get those, and what you actually are doing in your yard that might also affect what pollinators we're seeing there. Some things that we get a lot of questions about are, oh, is it okay if I water? And like, yes, you want to water to keep the plants happy, but if you're overwatering your garden, then that might actually be bad for bees. And so things like this, we want actually you to record for us after you register your garden. So we have a series of questions that we're asking you with your registration to tell us a little bit about how you take care of your yard normally, what other plants you have in it, whether or not you use pesticides or mulch or other things. We don't necessarily want you to change anything. We just want, these things are important for us to understand what patterns we're seeing. The last thing is, we did ask whether or not you'd be willing to have a scientist visit. That's not at all required, and it may or may not happen depending on how far away you are from us. But some of this actually helps us validate our data. It helps us compare what you're finding in your yards to what we find, and it serves as kind of a benchmark to see how similar the data is. And this is pretty standard with citizen science projects, because we want to be able to make sure that the data that we're getting is robust, so that we can, when we make our recommendations, we feel confident. So we're asking you to, after you have your bed set up, we're asking you to collect data at 4 different times. These are all the 3rd weeks in throughout the summer months here in Illinois. Starting June 17th, which is actually National Pollinator Week, so we thought that would be a great time to kick it off. July, August, and then in September. If you are someone who really gets into this and you would like to record data more often, please feel free, go for it, have fun. These are just dates that we're trying to target. And because one of the things we really struggle with as scientists is reducing variability in our data and all of these unexplained factors, and doing it on a similar week and having more data points collected around the same time really helps us do that. So during that week, and if it has to go a little bit before, let's say you're traveling, or a little bit after because the weather is bad, please don't stress out about this. We're totally flexible. If you can't get it done exactly June 17th through the 23rd, we still want your data. Please make an effort to collect the data, especially once you've gone through the trouble of setting up this bed. So during each week of surveying, we're asking you to fill out a floral survey. That is actually just a survey of your garden. One of the things that can be really important to know to attracting pollinators to these ornamental plants that we're trying to assess is what else is in your yard. If you have a huge, big, beautiful yard, if you have a small yard, but it has a lot of flowers, all of these things actually make a big difference. So we're asking you to fill out a short survey about the kinds of other things that are in your yard and how attractive they are to pollinators. We're also asking you then, so then you have the option, I'm sorry, of doing a Monarch survey, which is, David will talk a little bit about that in a moment, or doing these pollinator visitation surveys. So I'm gonna go straight to pollinator visitation surveys. So for these pollinator visitation surveys, we're gonna ask you, for the floral surveys, this is the initial survey of your garden. We're asking you to estimate your neighborhood of floral abundance. So whether or not, if you have a really beautiful garden and your neighbor also has a really beautiful garden, that can really influence the results we're seeing of the attraction of the ornamental plants. And so we're just gonna we have some simple questions to ask you, what does your neighborhood look like? Do you have a lot of neighbors that have really beautiful gardens, or are you the only one in, surrounded by cornfields? And that is obviously going to have a huge role in how many pollinators we see on some of these plants. And so we wanted to try and account for that a little bit. How many flowering plants do you have in your yard? Are you someone who plants only roses? Or are you someone who has a lot of different diversities of things? All of these things matter to pollinators quite a lot. And then lastly, we're trying to really get to a point where we can assess other plants in people's yards. So what flowers are attracting pollinators? So that's pretty important. And all of this is laid out again on the website. There's a data sheet, and we just are asking you to record that once a month. Then once you move on to doing your pollinator visitation surveys, we're really interested in whether or not you see bees, butterflies, flies, or other things on any of these plants that we've asked you to put in. We want you to pick 2 plants of the same variety. And then we're asking you to look at those 2 plants for only 3 minutes. Now, this is a really common method that we use it as scientists to record how attractive a plant is. It's just watch it for a very short period of time and see what comes to visit, and then write down how many things you saw visiting those 2 plants. And that should really help us get a sense of whether or not a plant is good or bad for pollinators, right? And so one of the things that we want you to be aware of is that distinguishing pollinators actually can be really difficult, right? Bees and flies can look really similar, and bees and wasps can look really similar, and flies especially can be super tricky. In the 3 photos that I've put up here, only one of those is a bee. And if you're thinking it's the yellow and black one, you would be wrong. That is a fly. So it's this green one at the bottom with the stripes that's actually is actually the bee. And so a few key things to look at is that both bees and flies come in yellow stripes, blues, greens, and oranges. The easiest way to distinguish them is that bees actually have longer antennae and more distinct body segments, and flies have like really huge eyes compared to their head size. Now, bees and wasps also come in the same color varieties, but for me, I always see that wasps look really mean. They have thin waists and they tend to have very few hairs on their body. Bees tend to be hairier, and oftentimes if you see something if you see a bee, I wish I had some videos, and actually we do have videos on our website that'll help you with this. If you see them moving around a flower, kind of collecting pollen, those are obviously gonna be bees because neither wasps nor flies collect pollen. Here's a more close-up shot, and there are a lot more identification resources on the website, and some short videos and diagrams as well. So these are the very large eyes I was talking about on flies. It takes up basically their entirety of their head. Bees have more obvious body segmentations, and they do have 4 wings instead of 2, but that's really hard to see on a flower. And then we're just asking you to submit your data. There's an online form to do so. but you can also submit it by mail if that's the way you would prefer. And we have recorded some information of those that are wanting to submit it by mail. So I'm super excited to have all of you participate, and I thank you so much for listening in today and for participating in this project.
Next up, Dr. David Zaya is going to talk about the Monarch portion of the project. And this is Deborah again. We're going through 3 projects today very quickly. So this is a lot of information, but to answer a question, yes, we will repost this if you want to go back and review anything, and all this information will be available on the project website.
Hello everyone, this is David Zaya. I'm going to be talking about milkweeds and monarchs. I'm actually a plant ecologist, but I'll start off with the butterfly. The monarch butterfly, our state insect, an amazing creature that has a lot of cool things about it, including how cool it looks. But it's been declining over the last 20 years. The monarchs in eastern North America migrate to Mexico in winter, and they overwinter there. In the area they cover in Mexico, its indication of how many there are has been declining quite quickly since the 90s. And a lot of conservation efforts in this region in Illinois and in the Midwest have gone into milkweeds. Milkweeds are connected to monarchs because milkweeds are the only larval food for monarch butterfly caterpillars. That is, the adults can eat nectar from all types of flowers, like not all types, but many types of flowers. It doesn't have to be a milkweed. But they have to lay eggs on milkweeds because that is the only thing their larvae will grow on and feed, so they can develop. Illinois is part of the summer breeding range for monarchs. Like I said, they migrate. The migration is just an amazing story, and due to limited time, I won't get too much into it. But they do this every year, 4 generations, about 2 generations, I'm sorry, breed during the summer months in the Midwest and Eastern United States. Then in about September, they decide, that last generation decides, I'm flying off to Michoacan in Mexico. They head down there over winter, and then once it warms up a little bit, just about a few weeks ago or a month ago, they start heading north. And right now they're probably on their way to Texas and Oklahoma. Although one of my coworkers saw one in Champaign, but it was looking pretty tired and probably not going to make it. So in this region, this is their breeding range. So that means egg laying and larvae. And a lot of the emphasis, like I said, is on milkweeds. And the goal for Illinois in the next 20 years is to plant 150 million more milkweed stems than what we have now. And my question, what I'm really interested in, is where? Where should these milkweeds go? Where would they be most helpful? I calculated that we could fit that in 1.5 sections, like a 1 mile by 1.5 mile wide rectangle. But that probably wouldn't do much good if we pop them all down in one space, right? So, but I really want to know all across the state, where do monarchs lay their eggs? Where are they most likely to use milkweeds? And where, once they lay eggs, do larvae survive? I'm interested in north versus south and east versus west, urban and rural landscapes, and then kind of at the smaller scale, milkweeds that are surrounded by other milkweeds and other nectar sources, or just pretty isolated. So this is how you can help. You are kind of hopefully helping me collect this data in a lot of different places where I couldn't be all at once during 1 week in June and 1 week in July and 1 week in August and so forth. And just to briefly repeat, where the milkweeds go in the garden, there should be 4 of them. They should go on the edge because you're going to get real up close and personal with them. The first data collection point is in the 3rd week of June. So as long as they're, say, about 5 or 6 inches tall by that time, they'll be big enough for monarchs. So I recommend you get them going before then. Unless you buy them large, then you can plant them the day of, and monarchs will lay on them the next day. I've seen eggs on milkweeds. And when you collect data, you'll be counting monarchs. It'll be eggs and larvae, or caterpillars, all types, alive, dead, damaged, all of them. You're going to tally them up. Then there'll be a separate section for dead and damaged larvae and eggs. And on the top, we have a freshly laid egg. And on the bottom left, you see an egg that's about to hatch. The caterpillar looks to be healthy and alive and is about to pop. And on the right, you see an egg with a dead embryo in it. And I don't know why that is, but we just wanted to record that. And it'll take some practice figuring out the difference. There are resources posted online to help you. And also you can see sometimes, I've seen this, like something small has come along and taken a bite out of the egg. So if you see that, just record it in the dead or damaged category. In addition to the egg thing, we need you to look at the larval instars. That is, the caterpillars of monarchs go through 5 stages between being an egg and a chrysalis. And those 5 stages are called instars. They molt in between each of those instars. And there are resources online to help you distinguish the 5 larval instars. And you don't have to get it exactly right, but take your best guess. If you get within 1 instar, that's still very helpful. And then we'll ask you to collect some simple things on the vegetation. That is how tall the milkweeds are compared to other plants immediately. Are they taller than the milkweeds? Are they shorter? And then how many of your milkweeds survived? We ask that you plant 4 of them, and yeah, we'll see how many make it. When you collect this data, look at all the milkweeds in one day. It should only take a couple minutes per milkweed. And we ask that you do it at least once per month in June through September with a minimum of 4 times all summer. And you can collect more than that. You can collect data up to once per week, but I wouldn't collect data more than that, more often than once per week. And well, we're going to ask you to do a little bit of homework to distinguish eggs from latex drops. So milkweeds have this milky sap that give them their name. And then sometimes that comes out in round drops that look a lot like eggs. And then the other thing is learning how to distinguish the different instars. Here are a couple of helpful resources, websites and groups that have some information, and we have information linked from those resources on our website. And also, just I'll mention the bottom one, the Monarch Labs MLMP, the Monarch Larva Monitoring Program. If you don't want to plant your own milkweeds but want to collect data on monarchs, that's a great resource to go submit citizen scientist data. And they've been collecting a lot of data for a lot of years, but they still need more, and they're answering some really interesting questions with that work. Here's the data sheet. So the main thing I want to point out is that each plant gets 1 row. So there's a plant 1 row, and you enter how many eggs and instar that are total, and then how many that look dead or damaged. And then once you move on to the second plant, you'll enter it there. This is a paper copy meant to help you with collection in your garden. But when you go to submit, we ask that you submit online, although mailing is okay. And then when you submit online, I'll just repeat something that Alex said. It's helpful to submit it as you collect the data rather than just dumping it all in October. It's okay if you dump it all on us in October, but it'd be helpful if we got it in a little bit of a stream. And I'll pass that along to Michael.
Okay, I'm Michael McKelvey. I'm here to talk about BeeSpotter. My colleague Terry Harrison should be on the line as well. He's remote, so we're going to see if we can get him talking. Terry, is your microphone working?
Hello?
Hey, okay. There's Terry. And Terry, so I'm Michael McKelvey. I'm the webmaster for BeeSpotter. And Terry, if you'd like to introduce yourself.
Yeah, I'm Terry Harrison. I'm the identifier and scientific coordinator on BeeSpotter.
Okay, and we'll be tag teaming this presentation, just going back and forth among the slides. And one quick aside before we start, every bee photo you're going to see in the BeeSpotter presentation was submitted by a citizen scientist.
All right, I'll start things off. We have this slide here. What is BeeSpotter? It is a citizen scientist initiative to engage the public in data collection regarding bee populations, housed here at the U of I, and it began in 2007. So we're basically interested in geographical distribution of bees and in seasonal abundance. Okay, how this started was that in 2002, the North American Pollinator Protection Campaign, which is a private group, requested a study from National Academy of Sciences on bee decline, because there was a lot of kind of anecdotal evidence. Everyone seemed to agree that there, on the whole, seemed to be a decline. But what the study did was try to find out, do we have good numbers on this? And one of the main conclusions of the study was that we don't. And so May Berenbaum, the head of the entomology department here at U of I, initiated this group, this citizen scientist-based group, initially just for Illinois, to see if we could generate some numbers on bee populations here in this state. And we started out soliciting photos just from Illinois, but fortunately we have a great bumblebee expert, Dr. Sydney Cameron, is in the entomology department right here. And so I had some conversations with her, and that in conjunction with some requests from people in other states wanting to know if they could participate. And Sydney informed me that the bumblebee species in these other states, in Missouri, Ohio, and Indiana, are pretty much going to be the same as what's in Illinois. So we were able to welcome records from those other states as well. And as far as the taxonomic range, we can identify honeybee and the 13 native bumblebee species from photos. So many of any other kind of bee are just so small and nondescript, it would be very tough. And if you look on the keys on the website, even for the bumblebees, some of the characters are like spacing of the eyes and things like, these are more designed really for having a specimen of a bee under a microscope. But what we use, of course, are the color patterns. So bumblebees are big enough and distinctive enough if they're in good condition with the color patterns, that if we get a good shot of one, we can identify it.
Okay, this is Michael again. So before we talk about how you can participate, we want to talk a little bit about what it is we hope that you would get out of participation in a project like BeeSpotter, including an increased appreciation of pollinators, learning to identify honey and bumblebee species, and also gain an awareness of differences in seasonal cycles and foraging preferences of honeybees and bumblebees, and increased knowledge about bees in general, increased awareness of the challenges that bees are facing, a sense of contribution to scientific research, and gaining an appreciation for the importance of accurate data collection and research. And also a sense of belonging to a community that cares about biodiversity. So in order to participate, there's essentially 4 steps to becoming a BeeSpotter. You first create an account on our site. You go out and you photograph bees. You come back to the site and submit your spottings. And then after an expert identifies them, you can see your contributions on the site. It is a photo-based participation, so it does require a camera of some sort, whether it's a point-and-shoot camera or a really fancy camera or even just your smartphone. And also some way to connect to the internet, either via your smartphone or via a computer in order to upload your spottings. You can read more about these steps for getting started at beespotter.org/getting-started. So the first step, creating an account. We try to keep it minimal, so it's not too onerous to sign up. So you just need your first and last name, an email address. You can choose a username, which we'll use instead of your name on the site, and also a password. Optionally, you can include how you heard about us, which is helpful just for our own knowledge of how people are coming to BeeSpotter. You are able to browse BeeSpotter and view spottings that other people have submitted without an account, but if you want to contribute to BeeSpotter, you do need to create an account.
Okay. So when you go out to photograph bees, and by the way, we do have a guide for taking good bee photos on the website. It should be pretty easy to find. But the idea is that each different spotting that you submit should be a photo of the same bee. And it can certainly be multiple photos because that often helps. Sometimes out of 5 shots, there'll be just that one that's absolutely perfect for identifying the bee. So we encourage multiple photos as long as they're all of the same bee within any one spotting. And as it says, multiple angles are useful. Again, what we're looking for is generally the color pattern on the bee. And yeah, if you get it, if you're, say the bee is on the flower and you're standing right above it and you're looking at its back, that's the single most informative view, especially if you can get it to get its wings out of the way. Sometimes certain individual bees seem to want to fold their wings over the abdomen as soon as they land, and that can make it difficult to see the color pattern. I think some of our photographers are able to talk to bees because they always seem to get them to spread the wings perfectly so I can see the colors. I don't have that good of luck when I try it, but so that's the general idea. And as it said, side view sometimes can be useful, and occasionally head-on view. So the basic idea is just like any other photograph, you would want it well lit. You would want the bee to be holding as still as possible. You can get as close as possible. When a bee is on a flower, it's working, and it's very mindful of that. And really the only time you're in danger of getting stung is when you get too close to the nest of these bumblebees. So it's not at all likely that the bee is even going to be interested in you other than maybe flying away. And certainly it's not very likely that you'll get stung. And we do prefer full-size photos because when I go to do the IDs, there's a button I can click to bring the photo up to full res. So, and that often is very helpful in seeing some detail that I need to see. And also, it's ideal if you crop the photo to where just the bee takes up the entire photo. I get quite a few photos in which initially I have trouble even seeing that there's a bee in the photo because it's a little dot right in the middle of a big, you know, bed of flowers or whatever. So that's not very good. The more bee I can see in a photo, the better.
Okay, and before I move on to the next slide, I just want to highlight the bee in this slide that is absolutely covered in pollen and one of my favorite photos that we've received. So the next step, once you're done photographing the bees, is to come back to the website and to submit your spottings. It's fairly minimal for each spotting. You just give it a title, which is best if it includes some helpful information for you to distinguish your spottings later. You might choose, where the location, prominent flower was on, the weather, something like that. What activity you were doing when you went out and spotted the bee. Also the spotting date, which may be different from the date you're uploading it on. That's fine. It's not always possible to submit the spotting the same day that you actually take the photos. The geographic location of the spotting, and we'll provide you with a zoomable map where you can just click to select where it was that you spotted the bee. Of course, upload your photo or photos of the bee, and optionally you can choose to try to identify the bee. You can try your hand at it, see how it goes. We have some guides to identifying bees that you can use to try to help you with the identification. But don't worry, if you get it wrong, we're still checking it over. We have an expert identifying it. So that's really just for your own personal use if you want to try it out.
Okay. As it says, last step here is that your bee gets identified. And this, you don't have to try to identify it yourself. And if you do, we hope you keep it fun. We do not keep any kind of metrics on how many you get right or how many you get wrong or anything like that. So anyway, once I identify it, then I send an automatically generated email to let you know it's been identified and what it's been identified as. Sometimes I'll put in a note about this is an unusual bee, or if you didn't identify it correctly, I'll note, okay, this one has 2 yellow segments on the abdomen, and the one you picked has only 1, so that's why it can't be that. So that's generally the idea here, is that once I identify it, then you'll be notified.
Okay, so we're running up against our time, so I'm just going to kind of skate through the next couple slides pretty quickly. The historical records at the Illinois Natural History Survey have 5,300 historical records over the course of over 100 years. So far, BeeSpotter has about 5,800 bee spottings submitted. And that's out of 66 attempted spottings. So we've had 6,600 records contributed, and 5,800 of them we have identified as bees, and that's over 13,000 photos. Some broad trends here that we have. Caveat, these are rough and broad trends comparing BeeSpotter data to the historical records. These are not scientific analysis, but you can see some differences that pop out between the historical records and the BeeSpotter data. In particular, Apis mellifera, which is the honeybee, makes up about 1/4 of our spottings in total. It was about 5% of the historical records. Bombus impatiens has seen a bit of a decline from 32% to 20%. Bombus pennsylvanicus has had a steep decline from 21 to 4%. We've actually had a couple, Bombus perplexus and Bombus rufosinctus, that weren't even in the historical record, and for which we actually have seen spottings. In particular, those 2 we didn't even have in our list when we started because we didn't expect to find them. We had to add them because people sent in spottings. And also interestingly, Bombus variabilis was 5% of the historical record, and it's the only bee we have yet to see.
Yeah, so here are the 2 unusual bees that we didn't even have in our list at the first. Both of these are found only up in northern Illinois. Bumblebees are odd. They have higher diversity up north, species diversity, than they do down here. So naturally, with the concentration of people up in and around Chicagoland taking a lot of these photos, it was only in our first full year, 2008, that the first Aphanus shown here on the left, showed up. And then it was 2011, I believe, when the first Rufosinctus showed up. So as I say, at the beginning, we didn't know if these bees were extinct in Illinois, but clearly they're not.
Okay, so there's a few different, there's various uses for the data so far, and including researchers, the Fish and Wildlife Service, data analysts, conservation groups, and many others. BeeSpotter data is collated with other data to help create range maps for where to find bee species and for scientists to help determine where to sample for their research. The Fish and Wildlife Service is also promoting BeeBlitzes around the Chicago area related to Bombus affinis awareness. We had a group use BeeSpotter photos for an image processing algorithm challenge to try to build an image processing algorithm that would distinguish between honeybees and bumblebees. And they used our photo set to train their bots. And then the photos that we get so many high quality photos, as you've probably seen throughout this presentation, that we get constant requests to use them for a variety of purposes, including in magazines, students doing school projects, conservation projects, conservation groups, and particularly interesting to me, the US Army's Entomological Sciences Division for their training materials, which apparently exists. So we have a few years ago started what we call a BeeBlitz. It's based on the idea of a BioBlitz, where all the biodiversity in a specific area is examined to provide a snapshot in time of the flora and fauna present. We're interested in what bees are present in one particular snapshot. This year, we're aiming for June 22nd, which is towards the end of Pollinator Week. And if you're interested in planning a BeeBlitz in your area, you can contact us and we'll highlight it on our website.
Okay, one last thing that's kind of as an adjunct to BeeSpotter is the Pollinatarium, which is out in the south part of campus. The building used to be the Bee Research Center, the U of I Bee Research Lab, which now has a newer facility on farther south. But the building was in pretty bad shape. And May Berenbaum, again, was instrumental in lining up a whole lot of volunteer work to renovate and refurbish the building. And people like Gene Killian over down at Paris, he was the state bee inspector for a long time, and he collected a lot of antique beekeeping equipment that he donated to it, and that's on exhibit there. And a local artist painted the outside of the building with these nice insect and flower murals, which are, of course, very appropriate. And inside, they have an observation hive of honeybees. This is just a full-fledged bee colony, only the walls are glass, so you can observe the colony. And there's also a prairie planting right on the grounds, so school groups go there, and like in the fall, they do these kind of meet the insects projects where they give the kids nets and let them catch this or that insect, and then they bring it up and we tell them something about the insect. And so, yeah, this school groups are a primary visitor to the Pollinatarium, and it's free. So I don't have any numbers on numbers of visitors, but I know it's a lot per season.
Yeah. Okay, and that is it. You can find us online at beespotter.org, Facebook, UI BeeSpotter, and on Twitter at BeeSpotter.
And you can also find the BeeSpotter link from the website you received in your emails. We're actually changing the URL. There's a different website in your email which you can go to now, but the new URL is what you see up here on your screen. And now we're gonna take time for questions. I see a couple of them came in during the chats. So we'll start with those. Let's see where our first one is here. One question, do all the flowers in the test bed need to be in one bed?
Yes, ideally all of the, when you set up a bed, it is one whole bed. Because one of the big challenges we have with designing this experiment or designing these gardens is making sure that they're similar across different people's yards. And if we have people separating them out, that actually makes it, that adds an additional element of variation, because then you may have, you know, one person's yard, they have their marigolds next to tulips or other, you know, or roses or other things in your yard, and another person's yard, they have it near something else. Having them all together, and ideally like a little bit isolated from the rest of your yard. It doesn't have to be totally isolated, but in a distinct area will help make sure the data is of a high quality.
And another similar question we had come in, Julie asked, what if I already have milkweed established without the other plants around them? Can I still report caterpillars from that milkweed?
That would probably be most appropriate for the MLMP program, the Monarch Larval Monitoring Program. I want, for my purposes, want to keep things as uniform as possible for the reasons Alex was talking about. Yeah.
Janet asks, for the past few years, I've been collecting monarch eggs and releasing adult butterflies. Do you want that information?
It'd be fun to share stories, but I wouldn't need the data. I've been doing that too.
Oh, and someone asked what kind of butterfly this is on the last picture.
Skipper, some sort of,
The skipper. I should know, I took that picture.
Oh.
Sorry.
A few things that I noticed while we wait for more questions to come in, a few things that as I was listening, that I either forgot to mention or wanted to mention again, is that one, for BeeSpotter, you can BeeSpot anywhere. It literally anywhere you are. If you are in a parking lot and you happen to see a cool bumblebee, go for it. You can also do your BeeSpotters in our gardens or in your own garden, literally anywhere. So if making a garden at your home is more of a commitment than you're ready for, maybe BeeSpotter is a great activity for you. But we really do hope that people will put in these gardens, because for me, for our project, being able to give people annual ornamental plants recommendations that are good for pollinators is really going to be critical to helping more people have pollinator-friendly gardens, for many reasons that some of the current recommended plants are really difficult to put in your yard. Like I mentioned at the beginning, my own personal story of like being fined by the city was more of a warning, but they did make me cut down a lot of my native plants that had gotten totally out of control. Honestly, I was not paying attention to them.
Hello, am I still on?
Yeah.
Hi, one thing I forgot to mention for BeeSpotter is that we do ask that you not submit photos of honeybees in an apiary. So honeybees on flowers are fine, but in fact, we've been asked by some beekeepers not to submit photos of honeybees in a man-made structure.
Right, and that's, this is Michael, that's kind of for the privacy of the people who are keeping the bees, so that we're not publicizing where they are in case somebody decides they want to go harvest and take away their hive. So that's kind of for their privacy.
Our Sangamon Extension Office asked, can you do one program or does it have to be all three?
I think that because David and I have similar objectives of wanting to look at specific plants, if you're gonna do the beds, you probably can do both of them. They actually, I know it sounds like a lot of steps, but it should actually not take very much time, because the observations are only 3 minutes once you have the bed established. We only anticipate each sampling round to take maybe an hour, hour and a half across the entire week. So it should hopefully not take very long. But BeeSpotter, you can definitely either add BeeSpotter to that or do BeeSpotter separately. And if you don't want to, I mean, if you don't want to look for Monarchs eggs, then you don't have to. But probably if you're going to put in the garden, you can have, do two. It's like one and one. You can either do all three or two and one, I guess.
Another question, someone already has native plant beds and wants to participate in the program. Will that data be useful?
Absolutely, because one of the things we're interested in is how your neighborhood is affecting the other plants in your yard and how that's affecting the attraction to ornamental plants and things like that. And that's one of the reasons that we ask people to record information, is because some people have beautiful gardens that are all ornamental plants, and some people have beautiful gardens that are all native plants. And those things might influence what we're seeing on the ornamental plants that we're interested in. So absolutely, if you have space in your yard, we would love to have the data.
Mm-hmm. Yeah, so that's regardless of what your garden looks like now, you can put in a test garden and join the study. Another question is, I have a raised garden bed that I was going to use for this project. Is that okay?
Absolutely.
Yep.
I mean, any space that you have, it's only 24 plants, so if the space is about 4 by 6, that should be fine. Yeah, I don't see any reason why you couldn't use a raised garden bed.
The next question is, to participate, do you need to establish the recommended garden? And I think the answer for that, for the ornamental plant study, was that yes, you do want to put in the ornamental test plot, since that's pretty essential to the study. But if you have, if you want to keep your garden as it is, you can still participate in BeeSpotter. Is that right?
Yeah. And if you want to collect monarch data, again, to repeat myself, you can contribute to the monarch larva monitoring program, MLMP, through Monarch Lab, but probably not to I-Pollinate.
Next question from Pat. Where do I get milkweed?
I've been seeing them at more and more shops, and then at farmers markets in spring, and native plant sales. I don't know, what part of the state do you live in, Pat?
Do you still have Pat Glidewell on the line? If you're not sure, I will say if you're not sure where to get milkweed or good plant sources in your local area, you can always contact your local extension office, and your master gardener volunteers or your horticultural educators will be able to give you some advice on what's near you. Next question is for BeeSpotter. Would cell phone pictures be accepted, or do we need a high quality camera?
Cell phone pictures are fine. That's what I use when I'm going bee spotting. You don't need a high quality camera. I mean, if you have one and you want to use it, you'll get fantastic photos. But cell phone quality photos are absolutely acceptable for bee spotting. So yeah, whatever is most convenient for you, whatever you find useful when you go out spotting.
Mm-hmm. I'm seeing some repeat questions in here about different types of gardens that I think we've answered. You can participate in BeeSpotter with any kind of garden, and in the other Monarch monitoring project that David mentioned. If you want to join the ornamental plant study, then we do ask that you put in that plant design. And I see some tips in the chat box here for where to get milkweed. So thanks for that, everybody.
If you start them from seed, you'd have to start them today and take real good care of them for them to be big enough on time. It's possible, but time's running out to start them from seed.
So I know possibly not everybody on the call has access to the chat box. So if anyone needs to call out a question, feel free to do so.
Yeah, so someone asked if they can't find the varieties that we listed, can they still use annuals of the same color? Yes, we do ask that that's why we came up with 12. So we do ask that you have the same species and color variety. So outside, I mean, hopefully those 12 are ones there. We did pick a lot that are very commonly selected or available in gardens. So hopefully there's something in there of those 12 that you can find. And just do your best, right? Like, it's not going to be perfect. We are covering a large distribution, and what's available to everyone is going to be a little bit different. But we do ask that you try and stick at least with the, the yellow zinnias and the yellow marigolds and things like that, that we hope to be, so that there's at least some similarity across the state, even if we gave example varieties, but if do not worry about that at all, it was just an example. Another thing that I forgot to mention was that for each plant species, we do ask that you collect 3 to 4 observations on them. So each observation, remember, is 3 minutes. And so we do ask that you repeat that observation a few times, just because having one single observation on one single plant in one single yard obviously is not going to be very robust. Maybe it's not a great day, or it's late in the afternoon or otherwise. The other thing to keep in mind is that pollinators like the same kinds of conditions that we like. They like sunny days when it's not too hot. So we ask that you try and focus on recording pollinators in decent weather, and maybe not during the hottest part of the day. So, in the morning before 11 or so, or 12, and then maybe late afternoon after like 3 or 4 o'clock.
Another question. Wildlife Refuge is asking why the Monarch project prefers swamp milkweed over common milkweed.
Swamp milkweed survives better than common milkweed, even in dry areas. I learned this the hard way last year, where I planted 1,800 plants, and about half of them died. And the swamp milkweed survived at a much higher rate. And it was just bad luck that I put them on the ground in June, and then it got really dry and hot immediately afterwards. So that's why I suggested swamp. Also, monarchs lay eggs on swamp milkweed more than they do on common milkweed, so that's also part of the consideration.
Janet asks if the online instructions will have all of the details for collecting data, such as time, number of collections, etc.
Yes.
Yes.
All of this. Katie, who has not spoken today, but is like the mastermind pulling all the puppet strings in the back. She has designed some beautiful data sheets and instruction sheets and a website and everything that should have everything that you need. So yeah.
And actually, this is Katie. There's also a question form on the website. So if you have questions as you're going along, please feel free to ask those.
Yeah, so I think all or most of the folks on the webinar, if you signed up, you should have gotten the note this week that the website has been updated with the latest instructions. And if you didn't hear, there is a question form. If you don't understand anything, just get in touch and we'll help you out. Colleen asks, can we make our test garden bed with more than 6 annual varieties?
Oh, that's a good question. If you're going to have more space, then sure, I think it would be fine. But we would like it to make sure that you have at least the 4 plants of at least 6. So, and I think getting more than 24 plants in that amount of space would be difficult. So I'd rather you have fewer varieties than more varieties and fewer plants, if that makes sense. Because if you have one plant of one thing and then another plant of another thing, it makes the data a little bit more difficult.
And you need 2 plants in order to do the
Right.
observations.
Yes.
And if plants
Yes.
die, you want backups.
Yeah.
And remember, this advice is just for that little 4x6 test plot. You can still plant absolutely whatever you want in the rest of your garden. But for the sake of standardizing their study, there was the design for it. Another question about milkweed. Would other milkweed varieties such as world milkweed be acceptable?
Some are and some are not. World milkweed gets many fewer monarchs on it than common milkweed and swamp milkweed. It gets about 10% as many. So I would not recommend that species. The main ones that you're going to find that are common in swamp, but also things like butterfly milkweed will get monarchs on it, and Sullivan's milkweed if you're up in the central Illinois region also is commonly will have monarch butterflies on it. But world is the one I would avoid, and some of the non-native ones I would avoid too because they cannot support monarchs.
So feel free to plant these anywhere else in your yard, but we're really hoping for our garden beds that you'll stick to the list of things that we've given already.
The question, can we use 2 varieties of milkweed in the garden bed?
I ask that you have 4 of one of the species and that you send me data on those 4 plants. But if you had, say, 8 plants and you had 4 common milkweeds and 4 swamp milkweeds, I have no problem with that.
Someone asks, says she has established common milkweed in her yard. Could she put the other plants around it?
Sure.
Yeah, that would be fine.
I think as long as it's not part of like a big garden that has a bunch of other plants, I think that's
And same person, can I report on 2 different test plots?
Sure, go crazy. I'm planning to put one of these in my yard as well. So, well, you know, if you have 2 different test plots that you would like to put in, please feel free. Go ahead, have fun.
Would they need a different
So-
address? or-
So yeah, for
Oh,
each
that's
bed,
true.
you have to put the address of that bed. So you should probably, don't put multiple beds in your yard. I think that's just
So is that
overkill.
confusing?
But if you're doing one at home and then you're part of also like a garden community that sets one up, that's fine. But obviously put the different addresses so we know they're different beds.
I forgot about that. That's how we keep track of them is through the address of where the garden is. So that makes it a little difficult.
Right, so yeah, we don't have a way to identify multiple beds at the same
Oh yeah, so
address
that's what
yet.
she said. One in my yard, one at school. So yeah, as long
Okay.
as the addresses are separate, that's fine.
Oh, perfect.
Uh-huh.
Another milkweed question, will butterfly weed work instead of swamp milkweed?
Yes, it will.
Okay. Butterfly weed is on the good list.
Although we don't have a good way to. I don't know.
There's another spot on the data sheet where they can
Yeah.
put the
Oh, okay.
So
species that they use.
if you don't use swamp milkweed, you can list the other species that you use. That's fine. And if you have a question about whether a milkweed will host monarch successfully, you forget about it, you don't know the question until later, you should email me. My name is David Zaya, and I think my contact's somewhere on the internet.
It's the same. So yeah, someone asked about Asclepias tuberosa. That's the same as butterfly milkweed. So all of those things apply. So yes, you can use them. Ideally, you would use swamp milkweed, but I know Asclepias tuberosa is super common and very easy to get. So if that's one that that's all that's available, then please feel free. It's not as good of a supporter, I guess, as the others, as swamp, but it'll do just fine.
It's about a half or a quarter as good, but it's good enough to get data from.
All right, we're about 10 minutes over time on our webinar. So unless there's any last minute questions, remember you can always reach out to us after on the project websites have contact information. You can contact me, Deborah, and I'll try and get you to the right place. But thanks so much, everyone, for joining
Thank you,
today.
everyone, and we really appreciate it.
Thank you.
This data is going to be really amazing for helping improve conservation efforts in our state for pollinators, so thank you.